Welcome back. So this is the 12th session of the part 1 of the series of lectures in Nonstationary Time Series Analysis with modern signal processing techniques. So the focus or the target in this session will be more applications of synchro squeezing in real-world data, and we will focus on, again, these 4 different types of missions. The first example is the decomposition capability to recycle cardiogenic artifacts. So this is a simulated data to show you, to help you to visualize what's going on. So this is a recorded airflow signal that you may see in practice, and this is the simultaneously recorded breath. This is a breathing activity inside the signal, and this is the cardiogenic artifact. And you can see that's the ECG signal, which has bit-to-bit correlation. Suppose I give you this signal, the question will be, for this airflow signal, our interest is the airflow, not the cardiogenic artifact. And that is why this guy is called artifact. So how can we recover this signal from the given airflow signal? That will be the interest. And this is the result. What do you get from the recorded signal, airflow signal? That is what you can get using synchro squeezing reconstruction, the hemodynamic signal, which oscillates with high correlation with the simultaneously recorded ECG signal, which tells you that reconstruction is good. Traditionally, what people did was using bandpass filter, and this is a result bandpass filter. You can see it doesn't oscillate very well. It has very strange behavior. And the literature is in this paper, which you can take a look if you have interest. And it also takes the D-shape into account. And this is another example. This is the signal recorded during the bronchoscopic, sedative bronchoscopy examination. This is the end-tidal CO2 signal. This is the respiratory signal recorded during the procedure. You can see a lot of spikes. And those spikes, small spikes, they are cardiogenic artifacts. And this is a decomposition result, which can help you to better identify the anesthesia dynamics. And this is the literature. If you have interest, you can go into details. I will not tell you clinical clinical details. And what I want to tell you is instead, this kind of recycling information is actually can be useful as a surrogate information. For example, in some situations, maybe the ECG signal is broken, but your respiratory signal is well, and it has cardiogenic artifact. And in this scenario, we can use the cardiogenic artifact to provide heart rate information when ECG signal is broken. And this situation is actually maybe not so common, but when it happens, it may be something that can help to save a life or something. Okay, so this is a kind of hypothetical scenario. Just let you know, it is why we call it recycling. And this is a surrogate heart rate information summary statistics using D-shape or traditional synchro squeezing. And you can see that if you use D-shape synchro squeezing, the result is actually better than the traditional synchro squeezing or even the traditional high-pass field or band-pass field. Okay, the second application will be the quantification of anesthesia depths. And you may recall this slide about heart rate, which is a portal to physiological dynamics. You get ECG signal, you get instantaneous heart rate, which is highly non-stationary. And if we can quantify it, it is heart rate variability. It can give us a lot of clinical information. And heart rate variability can be viewed as a lens or scope to understand your health from a different angle. It actually models our body we can model our body as a complex system, and the autonomic system is a key ingredient to integrate the whole system. And this autonomic system is reflected in the heart rate. Okay, so the heart rate variability in the end reflects part of the complexity. So analyzing heart rate variability reveals the complex mechanism from a different angle. Also, just like we are peeped into this complicated system from a different and so on. It is a very hard topic to quantify heart rate variability. So this is the slide, or not slide, the snapshot I took in 2023, December 28th from PubMed, which is the main database for medical research hosted by NIH. You can see there are kind of almost 60,000 papers results, and it starts to get hot since 1960. So that's a very active research field. And if you go to Google Scholar, you can see it is even 3 million. It's a lot of people, a lot of relative topics, and so on. Very hot topic. And we will discuss heart rate variability in the surgery and anesthesia. So this is a slide I have shown you. The focus is we want to study your brain dynamics during anesthesia, but we want to better understand the subcortical activity. Okay, instead of the cortical activity. The cortical activity will be studied using from EEG signal, which we will study in a bit, or maybe mainly in part 2, not in part 1. But anyways, so this is also a slide I have shown you, right? This is the instantaneous R. This is the R interval time series, and one of it will be the instantaneous heart rate. You can see a lot of dynamics inside. This is the synchro squeezing transform, and you can see, oh, this part is very concentrated, regular. And this part is sporadic, irregular, and this part is in between. So we can quantify how concentrated the signal is, and this is called the non-rhythmic to rhythmic ratio. And the idea is very simple. We just focus on the very dominated curves as one, or as the rhythmic energy, and the remaining as non-rhythmic energy, and take the ratio. That's it. And then this is the index you can get for the NRR. For details, you can go through the paper and see how the index is defined. And this is the clinical result for using this NRR, which helps us to better predict the first reaction. And this PK value definition is in below, but you don't need to worry about it. You can just imagine that if it is close to 1, it is a very good predictor. If it is 0.5, then it's just a random guess. And you can see that at time 0, this is when a subject wakes from anesthesia. And if you focus on the NRR index here, you can see we can very early predict the awakeness from the heart rate variability. Okay, but if you use BIS, which is the brain wave from cortical activity, you can see that it can predict or get the result only right after, or maybe very accurately after a patient wake up, because after wake up, the cortical activity increases and so on. And the other indices, heart rate variability indices, may not be super useful. That's the conclusion. Okay, and another application will be the cardiogenic artifact detection in the EEG signal, which has many applications in other kinds of artifact detection. But I will use the EEG signal as a motivation and and show you the result. And what you see is this is the recorded EEG signal from a subject sleeping. And you can see there are several spikes. And those spikes coincide with the simultaneously recorded ECG signal. But this kind of cardiogenic artifact may not always exist. It may sometimes exist, sometimes doesn't, which depends on how the patient sleeps, the position, many mechanisms. And as I mentioned before, at this moment, we do not fully understand the mechanism of cardiogenic artifact, but this is not a concern today. The question here will be, first, is there an artifact Okay, for example, I give you a segment of signal. Is there a cardiogenic artifact? Or I give you an 8 hours recorded EEG signal. When do you have cardiogenic artifacts? Second, if you have a cardiogenic artifact, then how to remove it? Because our interest is the EEG signal, but not the cardiogenic artifact. But the second question will be answered in part 2. In this part, we will answer the first question: how to determine if there is a cardiogenic artifact. The answer is very simple. We use synchronous squeezing again. So you have 2 EEG signals. Here you have very obvious cardiogenic artifact. On the left-hand side, you don't. So what you do is you do have some rectification, and you run synchronous squeezing. And the purpose of rectification I have mentioned, we want to emphasize, strengthen the fundamental frequency component, but we do not know why fully yet. As I mentioned in the previous session, it's an open question. Then we detect peaks, local peaks. You can detect local peaks, even local peaks. And once you get the peaks, you focus on the frequency concentration in the frequency axis at the location of the peak. And you can determine some features. And those features, we can train an SVM model and combine with experts' annotation of you have or you do not have cardiogenic artifact to predict cardiogenic artifact. And this is the result. Actually, the overall accuracy and F1 score are reasonably high, and to our knowledge, our work published 2 years ago was the first effort in this direction, and the result I think is quite reasonable, and we can use it to remove the cardiogenic to detect cardiogenic artifact and even remove it. And this is actually something we encounter in other studies. For example, with Jira in this EEG study, some cases may have cardiogenic artifact. If we can remove them, the cardiogenic artifacts, then we can achieve a higher accuracy, and so on. And it also provides surrogate information for our peaks. And this is a comparison with the true ECGR peaks. So overall, it's I would say it's quite good, and the median absolute error is 1.24 millisecond, which is very nice. Okay, and then the third and probably the last application today is the prediction of different channels. The mission is very simple in the following sense. How to continuously record airflow signal for a long time. So there are a lot of respiratory examinations that require highly accurate and bulky, complicated devices, modalities in a hospital. But during the pandemic, you know that you don't want to go to the hospital, right? Because it is a place with many possible problems. You want to stay at home. But at home, you do not have these bulky devices. Then what can you do? So a very natural question is, suppose we are able to easily record your thoracic movement or abdominal movement. Then can we predict or reconstruct the airflow signal? If we can, then at home, we may not need a bulky device to record airflow signal. We can simply use the chest band or abdominal band and get the desired information so that physicians can remotely give you some information or advice. But why is it possible? So if you look at this picture, okay, the abdominal breathing actually encodes the information about your diaphragm movement. When you breathe, your chest, your rib cage expands, your diaphragm decreases. And the decreased diaphragm can be captured by the abdominal movement, and the thoracic movement can be captured by the thoracic band. So you can have thoracic band, abdominal band, and they can capture different amounts of information. And now you know that the airflow signal we have, we can record it from our nasal, actually includes how much air is is exhaled and how much air is inhaled. And the abdominal, the diaphragm decreases will give you the height, and the thoracic movement will give you the area, the cross-sectional area. So in total, if you multiply these quantities, you can get a total volume of air. So it tells you that by recording thoracic movement and abdominal movement, we may already have some information about your airflow, but the relationship can be very nonlinear, So what should we do? So the proposed idea is very simple. We decompose the signal into different components. This is abdominal signal into harmonics. And for these harmonics, we can design some features. And these features, turns out we can assume to be located on some manifold, low-dimensional manifold. And we will systematically discuss this manifold idea or manifold learning in part 2. But right now, we just assume that we have some nonlinear structures, nonlinear relationships in between the phases, amplitudes, and everything of different harmonics. That encodes how you breathe from the abdominal perspective. Now, you can take thoracic and abdominal movement and you get features, right? Suppose we are done, and we have a simultaneously recorded airflow signal. We can view it as a predictor. Then what we want to do is, we want to predict the airflow signal using the encoded features in the thoracic and abdominal movements plus some noise. And this F is very nonlinear, and in general we don't know what it is. But the overall goal is we want to learn this F so that we can use the label data which you recorded from your thoracic and abdominal movement and simultaneously recorded airflow signal to predict when you do not have airflow signal but only the thoracic and the abdominal movement. What can we do? How accurate we can achieve? And we proposed, actually in this work, the paper is actually here. This is the paper. If you have interest, you can go into details. What we propose to learn this F, this function, is using Gaussian process regression. And this is the result. This is of the high quality reconstruction. This is low quality reconstruction. So the signal quality of the respiratory signal also plays a role. But no matter what, you can see that if the respiratory signal quality is high, we can recover it quite well. And there are several information. The black one is the ground truth, and you can see there is a band. This band is the uncertainty quantified by Gaussian process, and so on. And this is the overall result, the ratio of root mean square error reduction. You can see that in most cases, the prediction error is quite small, roughly less than 0.1. But sometimes it can be very low, in particular, when the respiratory signal, the airflow signal itself is very chaotic, then the signal quality is low. Okay, so it tells you that by combining synchro-squeezing as features and use Gaussian process as the learner, we can achieve quite a lot. Okay, and the more information, more results can be found in the paper. The last one, yes, this is the actual last one, but this is actually not just an interesting observation and an interesting recording. Just for fun, I don't have any conclusion from this one. It's just to show you. Because this EEG recording is very interesting. When I saw it, I said, what is this guy? You see, there's the ECG peaks, and there are a lot of high frequency fluctuation. What is it? And your first reaction will be, hey, oh, I know. This is just the power line interference. But turns out, no. Power line interference, 60 Hertz, has been removed. You get 20 Hertz, 40 Hertz, 80 Hertz. I have no idea what they are. And I have asked around, maybe it's aliasing, we don't know, we have no idea. But if you look at a synchro squeezing transform, you see 20, 40, 80, and this is not, it looks okay, right? But the main interesting part is here. If I zoom in, You see, this is instantaneous heart rate and harmonics. ECG signal is very spiky, so harmonics, right? But at this harmonic, okay, the 20 hertz component seems to disappear. It becomes super flat. Okay, and then next harmonic, it comes back to capture the dynamics. So what's really going on? It seems that there are some kind of interference in between. This is roughly maybe 10th harmonics or something, and the interference. So you see, there's always some fluctuation, right? Some dynamics, but here it's super flat, and you cannot see any extra information about fluctuation, and there's no spreading, no interference. I don't know what it is. I think that is everything for this session. Thank you very much.